As part of an ongoing paleontological project in the Grand Canyon, researchers uncovered exceptionally preserved soft-bodied fossils of tiny creatures that lived 500 million years ago, providing the first glimpse of the incredible diversity of the animals that swam in the area. During the Cambrian explosion, the fossils include those of a strange mouthless worm that ate by filtering microscopic organisms from the water, a relative of today's penis worms with a bizarre array of internal teeth, and an extinct type of crustacean with some of the most elaborate chewing limbs ever seen in the fossil record. This is the first study based on exquisite preservation of soft tissues, says paleontologist Alan Collins of the Smithsonian's National Museum of Natural History. These fossils show us what was happening in the water column during this critical time when most of the major animal groups we know today appeared. Until recently, the story of the Cambrian explosion was largely based on fossils of animals with hard shells and skeletons that were well suited to preserve in the fossil record. The problem is that many of Earth's organisms never developed these structures, leaving us with a biased view of early life. As a result, we've had little insight into the forms and functions that existed alongside their hard-shelled relatives and even less understanding of the environments they inhabited. More complete fossil examples of soft-bodied animals do exist, but they typically come from places with harsh environments such as deep ocean trenches or mountain ranges, making it hard to tell whether they represent normal animals or just particularly resilient weirdos. Now, though, fossils from the Grand Canyon are shedding light on life during the Cambrian explosion in an environment that was neither hot nor cold, shallow nor deep, and full of plenty of tasty microorganisms. In other words, a Goldilocks zone, an area that was just right for life to flourish. The Grand Canyon is famous for its layered bands of red rock. Those layers are actually made of shale that settled out of the water over millions of years. The shale is flaky and clay-like, which makes it harder to extract fossils from than rock that is made of calcite or sandstone. And that means the fossilized remains of smaller soft-bodied creatures often get left behind. Previous studies of the Grand Canyon's Cambrian fossils focused on the vast quantities of hard-shelled animals in nearby formations, so no one bothered to dig through the shale. But a recent expedition turned that around. The team searched for signs of life in the very rocks that everyone else skipped over. They painstakingly picked through the clay in search of anything organic, and it paid off. They found many well-preserved specimens in tiny pebbles, which turned out to be fragments of soft-bodied animals embedded in mud that hardened like cement. Using acid and a toothbrush, they painstakingly cleaned the rocks to reveal the fossils inside. The newly identified fossils come from the same time period and the same place, the Bright Angel Formation, but are 3 times better preserved than the fossils found there before. What's more, they add significantly to our understanding of early life. There's the mouthless filter feeder. It's related to the sea squirt, a marine invertebrate that attaches itself to rocks or other hard surfaces and sucks up food suspended in the water. The team suspects that this creature did the same thing. Though its head was strangely bulbous and lacked a mouth, its throat was lined with a mass of tiny teeth arranged like the bristles of a brush. The teeth were shaped to direct water through its open mouth and across a mucus-covered sieve to trap microscopic organisms. The food was then passed down a groove to its single stomach, where it was ground into a goo and absorbed by the surrounding tissue. Then there's the penis worm. The fossilized specimen is so well preserved that the researchers could even make out its brain and gut. Penis worms usually have simple tubular bodies with a pair of hollow tentacles protruding from one end. Food enters through the center of each tentacle and travels to a central trunk region, where the tentacles meet. There it's ground up and sent to a sac near the worm's rear. But this species didn't eat in quite the same way. It seemed to use its mouth to suck food into a thick, flexible proboscis, a long tube tucked underneath its tentacles. Once inside the proboscis, the food brushed against a series of tiny interlocking teeth. With each bite, the teeth scraped and crushed the food into small pieces, pushing it farther into the proboscis, where it continued its journey to the main grinding chamber. From there, the partially digested food traveled down the worm's esophagus and into its gut. The researchers were also able to identify an array of bizarre-looking limb-like structures belonging to an extinct group of crustaceans called the narwhals. Narrow-eyed limbs are incredibly diverse, but one feature they all have in common is that they're jointed. Even today, the earliest known arthropods with unjointed legs are widely considered to be ancestors of modern crustaceans. And yet the narrow-eyed fossils from the Grand Canyon show the earliest evidence of jointed limbs ever found. Based on their shape and structure, the team believes these limbs may have looked like feathers. Each one was capped with a small claw and lined with rows of tiny teeth along its front edge. Like the limbs of modern crabs, the researchers think they would have been great at stirring water and creating currents to capture drifting food. But instead of being attached to the body with a single joint, these limbs grew out of 2 or 3 segments, meaning they could bend in multiple places. Some even had tiny, almost molar-like plates of tissue between the joints. When the crab-like creatures moved their limbs back and forth, those teeth would have rasped against each other, pulverizing any food they collected. The discovery suggests that the evolution of jointed limbs predates the Narao by at least 18 million years. It also shows that early crustaceans had a wide variety of feeding styles, from crushing and tearing to rasping and grinding. Finally, it provides the first glimpse of these complex limbs in an environment where not everything was constantly moving and churning. Previously, the oldest evidence of these specialized feeding appendages came from places that were pretty hostile for most animals, says paleontologist Joanna Wolf of the University of New England, who was not involved in the study. So it's really exciting to see them in the Grand Canyon because you get a sense of what was going on in a more typical environment. The Grand Canyon's exceptional fossils also provide the first glimpse into how different animals interacted with each other. Among the jaw fragments, teeth, and limbs, the researchers found a smattering of imprints left by tiny burrows. These traces of burrowing behavior came from animals that weren't preserved well enough to be identified. But by studying their tracks, the team was able to determine that these mysterious creatures were living in burrows that they dug themselves, and all of this came from rocks that most paleontologists would have ignored. This study shows that there are still so many things to discover, says paleontologist Matt Lamina of the Carnegie Museum of Natural History. Even in a place that's been heavily studied, we're finding things no one has seen before, not just because they're physically preserved, but also because they haven't been properly identified. What's amazing is that we don't even know what the half of it is. Going forward, the team plans to search for other microfossils in the clay-rich rocks. They'll also conduct additional field work in the Grand Canyon to find more fossils of animals with hard shells and link them with the soft-bodied fossils they've already found. Meanwhile, scientists like Porter hope that this study inspires others to look beyond familiar fossils and consider the rocks that everyone else overlooks. I think this is a paradigm shift in how we think about preservation. Porter says it opens up new areas of paleontology that were previously unconsidered.